All right, everyone, welcome. If you have a biology benchmark coming up, then you are in exactly the right place. We're about to dive deep and break down everything you need to know about heredity and biotechnology. Think of this as your ultimate study guide. Let's get you ready to absolutely ace this test.
So here's our game plan. First, we're going to talk about how nature shuffles the genetic deck to create variety. Then we'll crack the code on the rules of inheritance. After that, we'll see how your environment can actually flip genetic switches on and off. Then we'll look at how we read the DNA blueprint itself. And finally, we'll explore the incredible, real-world applications of biotechnology. Let's do this. First up, where does all this genetic diversity even come from? Well, you can think of it like this. Every single time a sex cell is made, it's like nature is shuffling a deck of genetic cards to deal a completely unique hand. And that whole shuffling process, it's called meiosis.
Okay, this right here is a classic test question, so listen up. What's the difference between mitosis and meiosis? Easy. Mitosis is basically a copy machine for your regular body cells. It's for growth, for repair. Meiosis, on the other hand, is that special process that creates genetically unique sperm and egg cells. So mitosis makes clones, meiosis creates variety. You absolutely need to know that distinction.
And that brings us to another key pair of words, diploid and haploid. Your body cells are diploid, which we write as 2N because they have two sets of chromosomes. You get one set from each parent. But the sex cells made during meiosis are haploid, or N, meaning they have just one single set. Why? So that when they combine, the baby gets back to that correct diploid number. It all fits together perfectly. Now, don't get bogged down trying to memorize every single phase of meiosis for your test. Seriously, just focus on the big picture you see here. The main takeaway is the final outcome. You start with one diploid cell, it goes through two divisions, and what do you end up with? 4 genetically unique haploid cells. From 1 to 4, from diploid to haploid. That's the whole point of the process. So how do those 4 new cells end up being so unique? It comes down to 2 really cool tricks. First, you've got crossing over. This is where paired-up chromosomes literally get tangled and swap pieces of their DNA. It's a genetic trade. Second, there's independent assortment, which just means the chromosomes line up totally randomly before they get pulled apart. Together, these 2 things guarantee that every single sperm or egg cell is a one-of-a-kind genetic combo. All right, let's move into our next section. Now that we have all this genetic variety, how does it actually get passed on? You know, it might seem totally random, but there are actually predictable rules and patterns that control how we inherit our traits.
And our number one tool for figuring this out is the Punnett square. It's basically a simple grid that lets us map out the possible genes from 2 parents and then calculate the probability of their kid inheriting a specific trait. It's all about playing the genetic odds.
And here is a classic example you will definitely see. We're crossing 2 heterozygous parents. Both are big B, little b. Now look at the results. The genotypic ratio, that's the actual letter combination of the genes, is 1BB to 2BB to 1BB. But the phenotypic ratio, what you can actually see, is 3 dominant to 1 recessive. Why? Because that dominant B allele masks the recessive B whenever it's present. But of course, genetics is rarely that simple, right? Here are 2 really important exceptions. In codominance, both alleles show up fully and separately. Think AB blood type. You're not a blend of A and B, you are both A and B. Then you have incomplete dominance, and this is where the traits actually blend together. So a red flower and a white flower make a pink one. And there are just a couple more patterns you gotta know. Some traits have multiple alleles, which just means there are more than 2 options out there in the population, like the A, B, and O alleles for blood type. And then there are sex-linked traits. These are genes found on the X or Y chromosome, and they often show up in males more frequently because, well, they only have one X chromosome to work with. Okay, so we've got our genetic code, but that's not the end of the story. Not even close. Let's dig into how the world around you can actually interact with your genes to decide which traits get expressed. This graph is a perfect picture of something called a polygenic trait. That's a trait controlled by many, many genes working together. Think about things like height or skin color. They aren't just on or off, right? Instead, they exist across this huge spectrum, and that creates this classic bell curve shape you see here, where most of us fall somewhere in the middle. It's a great visual reminder that most human traits are super complex.
And this leads us to a really critical idea. Your genes are not your destiny. Here's a great way to think about it. Your DNA is like a giant library full of recipe books. Your environment, everything from what you eat to where you live, is kind of like the chef who decides which recipes to cook today. It can activate certain genes or it can keep others silent. And this interaction is just incredibly powerful. I mean, the temperature of the nest can determine if a turtle is male or female. The pH of the soil can make a hydrangea flower pink or blue. And for us humans, simple lifestyle choices like diet and exercise can directly turn on or off our genetic risk for things like heart disease. It's amazing. So with all this genetic complexity, how in the world do scientists actually read the blueprint? Well, let's look at one of the most powerful ways we do that, a technique called DNA fingerprinting. DNA fingerprinting creates this unique barcode-like pattern for every single person, and by comparing those patterns, we can solve all kinds of problems. In a classic crime scene like this one, all you have to do is match the bands. You can see it clear as day. The DNA from the crime scene is a perfect match for suspect 2. Boom, case closed. And this exact same logic works way beyond forensics. The more bands or DNA sequences that two people share, the more closely related they are. It's how paternity tests work, and it's also how scientists map out the entire evolutionary tree of life, figuring out how different species are connected to each other. Finally, let's talk about biotechnology. This is where we take everything we've just learned about genetics and we actually apply it to solve real-world problems in medicine, in agriculture, and in so many other fields. So a huge topic in biotech is the genetically modified organism, or GMO. This chart really lays out the whole debate. On one side, you have these huge potential benefits. We can make crops that resist diseases, need fewer pesticides, or even have more vitamins. But on the other side, it brings up some really important questions about ethics, potential impacts on the environment, and, the long-term effects that we just don't know yet. The impact of biotech is already all around us, a lot of times in ways we don't even realize. In medicine, we use it to mass-produce insulin for diabetics and develop new vaccines. In farming, it helps us create crops that can survive droughts. We can even engineer bacteria to clean up massive oil spills. These applications are truly changing our world. All right, let's bring it all home. When it comes to your benchmark, these are the 4 absolute must-know takeaways. You have to master how meiosis creates variety, how to use a Punnett square, remember that genes and your environment work together, and know the basics of DNA analysis. If you've got these 4 ideas down, you are in fantastic shape. And all of this leads us to one final huge question to think about. The science is moving forward so fast, giving us this unbelievable power to literally edit the code of life itself. The question for your generation isn't just, can we do it? It's should we do it? And who gets to make that call? Something to think about. Good luck on that benchmark.